Hey guys, so this is going to be our next installment as far as genetics is concerned. I want to really stress something with you. The last set of lecture notes were on monohybrids, and um, the monohybrid story on how to do those is extremely critical. So what we're going to do today and the next few days is go beyond monohybrids. And then after that, we're going to apply a mathematical component to it. So don't worry about that. It's not major math. You need to know your fractions and percentages and ratios, but it's nothing major. But my point is that despite what I tell you for the next couple of lessons, you can always fall back on monohybrids. So monohybrids are critical. If you're not sure about how to do those things, email me, see me during Zoom on Tuesday, whatever the case may be, okay? I cannot stress that enough. So make sure you know how to do monohybrids and make sure you have the vocabulary down. So if I say heterozygous or homozygous or heterozygous dominant, or the word allele, dominant, recessive, you know what those mean. I cannot stress that enough. All right. So if you need a review, please do that before you move on with this lecture. All right. So the next level of Punnett squares are called dihybrids. Di referring to two pairs of traits. So when we did monohybrids, that's like crossing, say, blue-eyed with a brown-eyed person. Well, that's one trait, eye color. Even though it might sound like two, two different phenotypes, it's really one trait, eye color, right? Well, dihybrid is combining two traits. Let's say, for example, I want to cross a brown hair dimple person with a brown hair no dimple person. So we have two traits, hair color and dimpleness. So these crosses are going to be a little bit bigger, but I like to talk to you about this because it stresses a good point about the principle of segregation and independent assortment. Okay, so a dihybrid cross is two pairs of traits from each parent. Now, I want to remind you, I can't see you, so feel free to pause during this video. Okay, so it's kind of like putting two monohybrids together. That's why, if you know how to do monohybrids, you can always fall back on those. So, again, as I said earlier verbally, an example could be like eye color and earlobes for one person. We have two different traits, eye color and lobe type. Normally, if it's a monohybrid, it'd be one or the other, but now we're going to combine the two together. Di refers to two. So, again, to give you a big picture of this, the eye color gene, let's say hypothetically, would be found in an area on the DNA molecule, say from this person's mom and then from this person's dad originally. If we're doing a dihybrid, we're including another set of DNA molecules. Let's say, for example, there's a gene for earlobe type down here and down here. Again, this is all hypothetical, all right? Just kind of give you a graphic of all this. Remember, when you write down those alleles, those letters, those symbols that stand for a gene on a DNA, this is what you're referring to right here. This is what you're referring to right here. So if I say big B, little b, that means there's say a brown eye gene here and a blue eye gene here, for example. Both these DNA molecules here are homologous because of size and shape, but also where the information is stored at on that DNA molecule. Same thing here. So again, this is kind of a big picture that a lot of students just kind of forget about when you start doing these crosses. You'll be able to do these crosses pretty quick, but a lot of students kind of forget about what's actually happening in the background. So if I have the genotype big B, little e, I am representing a dihybrid cross. All right. So this is like this. Big B here and little b here. And the little e would be like having a little e here and a little e here. That stands for, say, attached earlobes. Again, guys, when you write this genotype, this is what it's symbolizing. But to write or draw all this out would be crazy. So we simplify it by doing a genotype. So how many DNAs are being represented here? 4. 4 are being represented here. How many genes? 4. Right? So how many alleles? 4. So we'll review that towards the end. So what does the genotype mean then? Well, it stands for the phenotype, doesn't it? So in this case, if somebody was big B, little b, okay, let's just do eye color only, then that would stand for being brown because brown is dominant allele, right? If somebody's little e, then that'd be a attached earlobe. That's a recessive trait. So the combination of these two means that this person is brown-eyed and attached earlobes. In other words, this person is heterozygous dominant for brown and homozygous recessive for attached earlobes. Okay, so get a little ahead of myself here, but I want to kind of give you the big picture of what's going on here, what it all genetically means. So let's do an example, all right? Let's do an example. So the last example was a human example. So this one is going to be a plant example because we can't forget about plants because plants do the same kind of process with genetics. Now, before we get into that, let me go back to this for a second. If I have two genotypes, all right, one is homozygous dominant, big B, and homozygous recessive, little e, and the other one is homozygous recessive, little b, and the other one is heterozygous, b, little e. You know, what does that mean? Well, if we're going to cross two parents, right, what that means is we're going to take the genes that are found on 4 DNA molecules and cross them with another parent with 4 as well. So this is why it's called a dihybrid because we're talking about two different traits. Eye color, for example, and earlobe attachment as an example. So what would this look like as far as the words are concerned? I'm going to cross homozygous dominant for brown eyes and recessive for attached earlobes with homozygous recessive for blue eyes and heterozygous for free lobes. So again, instead of writing all this down, it's easier to put it in code. So we're taking the wordage and converting it into code, into a genotype. This is why it's important you need to know the vocabulary. If I say homozygous dominant, what does that look like? As an example with bees. Or if I say heterozygous blue, you know, what does that look like? Homozygous recessive for blue. What does that look like in code? So there's going to be some times where I'm going to give you just a genotype, and sometimes I'm just going to give you just the words, and you're going to have to convert the words into a genotype. Okay? All right. So this is the big picture. You guys will be worrying about the code and the words. But remember, each one of these alleles stands for a gene on a DNA molecule. And these are homologous because the gene is located in the same place here as well as here. Homologous. Okay? Okay. So as I was saying earlier, what I want to do is an example using plants. So I'll give you a few minutes to write this down. Let's talk about watermelons. It's getting towards summer, so I thought watermelons would be kind of a nice one to do. Let's say I want to cross two watermelon plants that are both heterozygous for being green and small. All right, and by the way, it's going to be a bit of a mind game a little bit because just because it's a certain phenotype doesn't mean that's going to be a certain genotype. anyways, I'll get back to you in a minute. So, in the world of watermelons, green and small is dominant. We actually mentioned that a long time ago. The recessive versions of watermelons are striped and large. Striped and large. That's actually recessive. So again, I think I mentioned this to you before. Somehow, some way, I'll always tell you what's dominant, what's recessive in all these. Um, in this case, that's the watermelon story. So there are steps on doing a dihybrid. I cannot stress enough to you guys again. Make sure you follow the steps exactly as I have it on here. Just like for the monohybrid that I told you earlier, make sure you follow the steps exactly as you see these, okay? Do not deviate. Promise me. You do these 4 steps exactly as I tell you, okay? All right. So, step 1. We got to figure out what the parents' genotypes are. So, I gave you the wordage up there. Can you convert the words into code, into our genotypes? So this is going to be a diploid number. So for dihybrids, the diploid is going to be 4. Each parent is going to have 4 alleles. So this is if necessary. Today it's necessary. Sometimes I might just give you the genotypes, but today I'm giving you one of the worst case scenarios. Complete wordage. Okay? All right. So what would be the parents' genotypes? If you want to pause here for a second and think about it, go ahead. Okay, here are the answers. Now, you want to also choose an allele that best represents the trait, right? The best code. So for this example, I chose the following. I'm going to say, since green is dominant, I'm going to use G. And since S is dominant, I'm going to use, small is dominant, I'm going to use S. Now, by the way, how do I know that green is dominant based on this question, right? Well, it has to do with this term right here, heterozygous. Go back to your notes. You wrote this down somewhere. You cannot be heterozygous recessive. You can only be heterozygous dominant. So if I say something is heterozygous for being green, that means green is dominant. If I say heterozygous for small, that means small is dominant. So if I say heterozygous for brown eyes, that makes sense. I can't say heterozygous for blue eyes because blue eyes are recessive. You cannot have heterozygous recessive. There's no such animal for that, no such thing for that, okay? So you kind of pick up on that as you do more and more problems. So here's my scenario. Since both watermelons are heterozygous, each one is going to be big G, little g, biggest little s. Now, you got to have these in pairs. So the G's got to be in pairs, the S's have to be in pairs. Why? Because the watermelons got male and female, mom and dad seeds. So one came from the male and one came from the female of this watermelon plant. It's mom and dad. So we have to do these in pairs, right? You also notice I'm not mixing these things up either. I'll get to it in a minute. So the G's are on one side, the S's on one side. So you always want to keep one trait on one side and the other trait on the other side. Now, this one here is for the other parent. And the reason why I know it's the other parent is because it says both. Both are this way. So in some cases, I'm going to spread out the question, and there'll be one for parent 1 and one for parent 2. But for this one, they're both, so they're both going to have the same genotype. So before we kind of move on here, does the code make sense to the words to you? If I say heterozygous green, does that make sense? If I say heterozygous for small, does that make sense? All right. Are you putting a line over the little s? You know why? Because the big S, the little s look similar. I don't have to do that with my notes because you can see it very easily, but please get in the habit of putting those lines over those lowercase letters that look like the uppercase letters. All right. I personally think this is the hardest step right here. That's because you have to know the vocabulary. All right. All right. Step 2, draw the square. Now, because we have e-learning, you're going to be drawing probably more squares than I would typically have you do. Sorry about that. But if you do the math and figure all this out, the square is going to look like the following. So I'm going to have you draw this out. There it is. It is 16 boxes. Don't freak out. It's not that bad. I also want you to note something too, that the boxes are a bit more like rectangles. If you kind of see where we're going to go with this, each one of these boxes are going to have 4 alleles in it. 4 alleles. Okay. Step 3, figure out the gametes. Figure out the sex cells. Now, the answers to this will be half of what the parents are. So if the parents have 4 as a diploid, these guys are going to have 2. Now, this is done by meiosis. All right, this is done by meiosis. This helps to prove the principle of segregation that we talked about earlier in the notes. So the prophase 1 to the telophase 2, the making of the sex cells, that's what this is. I'm not going to go through all those phases. What you need to think about is, when I go through the steps of this, the phases of meiosis 1 and 2 are what are causing this to happen. Okay? So remember, when the chromosomes are being put in the center and separated, all that good stuff, the genes are going with them. And it's the genes that we are focusing on here. The genes that are represented by the alleles, by the letters. Okay, now again, this is critical. You must follow the steps exactly as I tell you, okay? All right. So a couple of rules. First of all, I want all answers outside the Punnett square. Keep the traits on the same side. You know, for example, in our watermelon, I want color on the left side and shape on the right side. So don't be mixing the traits up. All right, don't be mixing the traits up. Keep, in this case, color on this side and shape on the other side. Or if it was about humans, you know, keep eye color on one side and earlobe attachment on the other side. Don't mix the traits up. Very, very important. It's easier to read the answers if you do it this way. Okay. Well, guess what? We're going to employ a little bit of algebra here. So this is one reason for algebra, genetics. So I don't know if you've run across this before, but there's a process. There's a really easy way of figuring out the sex cells, in this case, seeds, you know, for our watermelons, by using a method called the FOIL method. And you want to do this for both parents. Now, since our parents are both the same thing, big G, little g, big S, little s, we really don't have to do it once. But if you have 2 parents, you really need to do it for both. If they have, especially if they have 2 different sets of genotypes. The FOIL method, right? Okay, so quick lesson on FOIL method. These stand for a certain order. All right. First, outside, inside, last. So when you're doing these notes, I want you to have a lot of space right here. So this is purposeful. Give yourself some space here. Now, I'm going to show you a couple ways of doing this. And I don't care which way you want to choose, whichever way you like to do it. In fact, by the time we're done with this, you might even be able to do it in your head. All right. But I'm going to ask you on an upcoming quiz to actually show me this process. All right. So here's the situation. Our first parent is big G, little g, biggest little s. The first thing I want you to do, like on scratch paper, for example, not here, but on scratch paper when you do actual problems, or in the space provided, I want you to take that genotype of that parent and I want you to spread it out, just like you see right here. Now comes the order. The F stands for first. The first what? The first 2 alleles of our traits. So that'd be big G and big S. So the first set of sex cell combinations will be big G, big S. Now outside. O stands for outside. That is the two outside alleles of the genotype, big G and little s in this case. So when you're doing these notes, I want you to physically do these brackets for me, okay? So you figure that out, and that answer then is big G, big S. You put it aside right there. I stands for the two inside. That's little g and big S. Okay, so again, do that as well in your notes. So that answer goes over here. Now, you notice I have a little asterisk right here. Here we have a bit of an exception. When you figure out gametes for Punnett squares past monohybrids, like dihybrids, the uppercase in front of the lowercase rule does not apply when you figure out the sex cells. So it's better to keep the traits separate on the same side. See how G is on the left side here, as I said up here, and the S is on the right side. It's easier this way. So this is an exception to the uppercase, lowercase rule, all right, when you figure out the sex cells for the dihybrids. Then L is last. These are the last two alleles of the traits. In this case, little g, big S. So then you write that down someplace. Now, I kind of prefer doing it this way. There are other forms as well I'll show you, but again, you choose which way best works for you. But guys, I cannot stress enough. You must follow this order exactly from left to right, FOIL. And when you place the answers down, place them on, along the side. Don't do it in a column. Just place them like horizontally. Please follow this exact process. So, what have we done here? We have created then all the sex cell combinations that could happen after meiosis. All right? If you remember, in meiosis, the chromosomes can be on either side where they separate. You might have a little bit of mixing up. So instead of trying to figure all that out, this is a nice, easy way of doing it by using this thing called the FOIL method. And so what we've done here is we've provided sex cells for all the sex cell combinations by using the FOIL method here. There are other ways to do the FOIL method. I've seen happy face version. I've seen angry chicken or mean chicken. I don't know if I really recommend these two, but I kind of like this version right here the most. So how you want to do it. So guys, if this was if this is the scenario, this big G, big S would be in this sperm cell. This big G, little s would be in this sperm cell. Little g, big S would be in this one. And little g, little s would be in this one. And since the other parent is the same thing, those would be found in their egg cells. So what we've done is we figure out all the sex cell combinations. And now we got to put these together. All right? Okay. So you then take those FOIL possibilities for each parent and you put them outside that square. You do not do it like this. No. Remember, the FOIL possibilities were two alleles, right? Haploid, like big G, little g. So when I see students do that sort of thing, that tells me that they did not do the FOIL and they don't understand the process. You got to take, a parent that's diploid, in this case, big G, little g, big S, little s, and figure out all the haploid possibilities. That's what meiosis is, right? Going from diploid to haploid. So the answer to this is this. So first thing I want you to do, draw another box. Don't hate me. Draw another box. It's 4 rows, 4 columns. All right. Then place the possibilities outside the square. So this is, for example, if this is the watermelon, this is like the pollen contribution. Well, the other one was the same, right? So we place those on this side. This is to be like the ovules, the ones that be found inside the flower and the ovaries. These are the possibilities there. So by doing the FOIL method, we figured out all the sex cell combinations of the genes. All right? All right. So what would then be the offspring? What would be the seeds of our watermelons, right? What would be all the possible combinations that can happen with this particular parent group? Well, now we move into our last step. Now we figure out the zygotes. We figure out the offspring. The answers are going to be 2N. So just like the parents, they're going to have a total of 4 alleles. This is proven by means of fertilization. You know, this pollen grain could combine with this ovule and make this seed. Or this pollen could combine with this ovule and make this seed. What would be all the possibilities, right? This also helps to prove the independent assortment principle. That just because you're green doesn't mean you're striped. You can be green and solid as well, right? You could be small and striped, small and solid. So color and pattern are independent of each other in the watermelon world. It's just like humans. Just because you're brown-eyed, does that mean you're going to be tall? No, because there's people with brown eyes that are both tall, medium, short, right? Independent. That's because all these alleles are found on separate DNA molecules, as I showed to you earlier. So because of that, we're going to be able to find the combinations here. All right, so some rules. All the answers go inside the Punnett square. Keep traits on the same side. All right, so one more time, don't hate me. I want you to draw us out one more time like this. And then I want you to take a few minutes. I want you to pause this video, and I want you to choose 2 or 3 combinations. So what you're going to do is you're just going to triangulate it. All right. So for example, you're going to take this one and combine it and put the answer right here in this box. Now remember, keep the traits separate. So the G is on one side, the S is on the other side. So go ahead and pause and go ahead and give yourself a few, try a few. Okay, so here are the answers. Now, did you do this correctly? Did you go from left to right, up and down? All right. In other words, you just pick and choose, or did you actually, follow it like a book? You want to do it like a book. So here are the answers. I don't know which one you did, so I'm going to give them all to you. You notice how I'm writing these down from left to right, up to down, just like how you read a book. You must follow that policy. Talked about that when we did monohybrids. So there we go. Those are all the possible gametes right there. Okay. So what would be the phenotypic results, or F1? So F1s are all our offspring right here. What's the phenotypic result? Well, we're going to have some green small, right? Now, by the way, how do I know this? You go back and you look at the original question. So if you have a big G, I don't care if it's 2 big G's or 1 big G and 1 little g, it's got to be green. And the same thing for small. Small is dominant. I know you got to think about that. If it has a big S, that means it's small. So we're going to have some of these in there. We're also going to have some green large ones in there. Here's a green large one right here. The code reads that way, like that. We're also going to have some striped small right here. Here's a striped small. I saw those in the grocery store the other day. And then we also see some striped large right here. Striped large. Again, how do I know that? I go back, look at the original question. It tells me what's dominant, what's recessive. So the law of independent assortment is proven with dihybrids, because you can be small and striped and small and solid. You can be large and striped, large and solid. Color has nothing to do with size, for example, in the watermelon world. Now in biology, it's not as easy as that. There are some exceptions. We'll get to that later. But for now, Think of it like this. All right, so here's our answers. Now, we have some ratio results here. When you do a ratio, upper left to bottom right. Upper left to bottom right. So, what would be the genotypic ratio of this cross? Well, how many big G, big S's are there? There's one, right? Now go down a little further here. How many big G, big S, little S's are there? There's two. How many of these? How many of these? How many of these? There's the answer. It's a 1, 2 to 1 to 2 to 4 to 1 to 2 to 2 to 1 ratio. That 4 right there are these 4 right here. So when you go from upper left to bottom right, you go from outside in. Here's our 1. Here's our 2. Here's our 1. Here's our 2. Here and here. Here's our 4. When you get past the middle, then you go from inside out. Here's our 1. Here's our 2. Here's our 2. Here's our 1. What about the phenotypes? Where am I putting myself here? Well, how many solid green smalls are there? Here's one right here. Here's one right here. Here's one right here. Here, 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 here. By the way, I know the pattern. I've been doing this for 20-some years. Gosh, I'm actually going on 30 years now. Anywho, so I know the pattern, so that's why I'm going through this kind of quickly. But if you have a capital letter of G or S, that is going to be one of these watermelons, isn't it? What about this one? This is a green but long one, right? And what about this one? That's a striped long one. And this one right here, that's going to be a long striped one. So, if you do the ratio for this, the answer is, you know, I may have said that wrong. I do apologize. Let's go back. Here it is. You're going to have 9 that are going to be short and solid. This right here, that big G, little s, that's going to be a long solid. Same thing here, and same thing down here. Again, how do I know that? Just look at the original question. This right here, that's going to be a short striped. Same thing here. Same thing here. And this one here is going to be a long striped. So the phenotypic ratio, upper left to bottom right, is going to be a 9-3-3 to 1. 9-3-3 to 1. I'll give you a little clue. If both parents are heterozygous in a dihybrid, it's always going to be a 9-3-3 to 1. Now, by the way, if you want to kind of check yourself on the ratios, does this add up to the total number of possibilities? Our total number of possibilities is 16, right? So these should add up to 16 if you did it right. It's not 100%, but it might give you a clue. Then there's some other questions I can ask you. For example, you say, what's the probability of getting a watermelon that's this? Big G, little s as a fraction. The answer is 1 out of 16. If I say, what's the probability of getting a, say, a striped short? That's these 3 right here. Striped short as a fraction. That's going to be 3 out of 16. See how I'm going with this? So I can also ask you other questions that relate to this. The probability of getting a solid small would be 9 out of 16. Then I might ask you to give me, a percentage. So if I want to know the percentage of getting a short, solid green watermelon, which by fraction is 9 over 16, you're going to have to take a calculator and divide 9 into 16. make sure you times it by 100 to give you that percentage, to move that decimal point over, and that'll give you a percentage. So you may want to have a calculator handy from this point on in these notes. All right, guys. That's kind of a lot right there. I'm going to go ahead and stop this video here, but I'm going to then show another video, and I'm going to show this video in the next video, because I'm not really supposed to go, really past 30 minutes and all this. Please, if you have any questions, email me. I'll be happy to talk to you about it, okay? All right, that's it for that. We'll continue on.